So, hey everyone, today we're going to talk about the function of antibody proteins. Antibodies are just another type of proteins, except the function of antibodies is to recognize and remove antigens, which are foreign body invaders. Foreign invaders could be like space invaders or like aliens from the deep depths of space, an exoplanet far away. And if somehow the whole Earth could come together and create a single army or something, they would represent the antibodies because antibodies remove antigens, which would be the space. So antigens come in many different forms, shapes, and sizes. So your body has to have a lot of antibodies to recognize and remove them all. So each antibody has this basic Y shape. If you get confused, just remember that this diagram looks a whole lot like a uterus with your basic fallopian tube, and I really don't know the rest. They're composed of 4 polypeptide chains, which are attached through disulfide bonds. The 4 chains come down to be 2 heavy chains and 2 light chains. The constant region, which is located at the base of an antibody, has an identical primary sequence of amino acids with all other antibodies. So for example, let's say we have antibody Bieber and antibody Timberlake. They would have the same sequence in this region, the constant region. But the variable region, which is located at the top of the antibody, while the constant region is located at the bottom, allows the antibodies to recognize different molecules. So antibody Bieber might only recognize Bieber molecules, while antibody Timberlake recognizes only Timberlake molecules. However, most antibodies are very specific, binding to specific molecules or specific regions on a specific molecule. These specific regions that the antibodies can bind to are called epitopes, E-P-I-T-O-P-E-S. Antibody specificity can be useful in the purifying of proteins from cell cultures. So let's say for giggles you have a culture tube full of all kinds of proteins, but you only want the Beyonce protein. You would use only antigens that would bind to the epitopes or specific regions of the Beyonce protein. A lot of times antibodies are used as commercial testing reagents or as test kit style things during research and manufacturing. Pregnancy tests could be an example of commercial testing kits that use antibodies to detect proteins and are a good way to see if there is blood in your oven. But if you can't afford antibodies or pregnancy tests, then you could use the enzyme-linked immunosorbent assay, also known as ELISA, or Lisa with the knee, or like, hey, Lisa with like 14 Y's or something. So in an ELISA, you have again your culture tube full of all kinds of proteins, and you want maybe the Taylor-Lautner protein. So you would test your sample with a special antibody enzyme complex that contains a special enzyme. So once the antibody is attached to the desired protein, the attached enzyme causes a color change in the reagent, which is then used to determine the amount of antibodies that binds to the protein. So the more the Taylor-Lautner protein, the more the antibodies do their thing, and then the more the color change and the darker the color. ELISAs are important in protein studies and manufacturing for this purpose. A special group of antibodies known as monoclonal antibodies are produced in cells made by fusing immortal tumor cells with specific antibody-producing WBCs, or B cells. This results in hybridomous cells, which grow and make large amounts of the specific antibodies that were coded for in the original B cells. The advantage in this process of monoclonal antibody technology is that many identical specific antibodies can be produced in large quantities. And so I know this is a lot of information to take in at once, so let's review. Antibodies strive to protect the human body from foreign invaders, known as antigens. Antibodies are made up of 2 light chains and 2 heavy chains. In the constant region, there is an identical sequence of amino acids, and the variable region is what differs the antibodies. ELISA, enzyme-linked immunosorbent assay, uses a special antibody enzyme complex. When the antibody binds, the enzyme causes a color change in the reagent to show the amount of the desired protein. Monoclonal antibodies are produced in cells made by fusing immortal tumor cells with specific antibody-producing WBCs, which are B cells. This results in hybridomous cells which grow and make large amounts of the specific antibodies that were coded for in the original B cells. So that's it. And I bet you thought it was me drawing the whole time, but it was actually me. And no, you're right. This isn't my voice, because this is my voice.